Assalamu alaikum. My thesis title is Progesterone Anabolic Steroid Microorganism Mediated Derivatives and Anti-Metastasis Drug Analogs against PC3 and HeLa cancer cell line. Myself is Sadia Sultan. These are my co-researchers. This project is about the development of the microorganism mediated derivatives of progesterone and anabolic steroids. Conversion of steroidal compound by microbial transmission is found out to be an important milestone in the research and development of steroidal drug industry. The chemists all over the globe are motivated not only for the environmental banning synthesis new products but also to develop green synthesis for the existing chemicals. The most important fact, the release of hazardous products of chemical industries and the release of agrochemicals in the atmosphere, land and water. Microbial transformation is a solution. It plays an important role in the production of commercially valuable steroids and terpenes. It is a bioconversion, a method of chemical alteration. It is usually selective and selective, and they are used for the biotransformation. This methodology includes the fungi isolate, fermentation, HPLC profiling, followed by spectroscopy analysis of biotransfer products, structure elucidation, finally the biological assay and the molecular dogging studies. Biocatalysts offer rapid synthesis of metabolite. It is a unique catalytic properties. Overall, is an biocatalysis increasing a pivotal technology in the toolbox of biosynthesis. These are my research papers, current research papers. Use of integer cells of biotransformation of steroid. Use of integer cells for biotransformation of steroid is well-distinguished process of recovering good yield of novel lead molecules. Here we use the megaestrogen acetate. It is an early active synthetic derivative used to treat the estrogen-dependent advanced carcinoma breast cancer. We have subjected this compound with Ganglion elegans for the first time, and two novel metabolites obtained from the epoxidation of megaestrol acetate. And another compound, testosterone propionate, also subjected, and is an important steroidal hormone responsible for the sexual characteristics, and it has been employed for the treatment of breast cancer. One new and four non-metabolites were obtained with testosterone propionate. These compounds were also subjected for biological assay and anti-cancer activity against PC3 cancer cell line and HeLa cancer cell line. And compound 2 and 6, as you can see here, found to be very potent, while the never been reported transformation compound 4 have moderate activity, and compound 3 and 4 also found to be active against PC3 cancer cell line. Oncotarget studies was also performed in order to have the microtubule targeting agent have an established history of utility in the treatment of cancer and have been instrumental as biological probes to identify the nature of tubulin and the role of tubulin dynamics in mitosis. Compound with anti-cancer property have the potential to inhibit the tubulin polymerization by binding to the colchicine binding site. Molecular modeling was also performed to understand the observed experimental activities and also to obtain more information on the binding modes and interaction between the biotransfer product and the enzymes. For compound 2, we can see here the binding interaction with the residue within the site of tubulin alpha beta dimer. For compound 6, also the binding interaction of compound with the residue within the active site of tubulin alpha beta dimer. This reference to marketing and commercial value. Here we take the example of cortisone production. Traditional cortisone was synthesized from the deoxycholic acid multi-steps, and high production cost $200 per gram. With the help of microbial transformation, we can cut down the step only 11 steps and reduce the cost $1 per gram. And with the help of this one, we have able to have two novel metabolites, 3 and 4, as an epoxidation of a double bond. Future plan involved the collaboration with the pharmaceutical company, Cancer Research Center, in order to develop new drug analog of testosterone propionate and megaestrol acetate derived to prevent the metastasis of MD/MBK cell line. This work will also help to improve the health of Malaysian citizens. As a conclusion, we can see the increased toxicity of compound 2, 3, and 6 in PC3 cancer cell line and their strong inhibitory effect against tubulin polymerization suggests that the combination of the strong hydrogen bond with hydroxyl and acetate group is desirable for biological activity. Experimental molecular doubling of compound 2 and 6 into colchicine binding site of tubulin suggests that these compounds have potential to inhibit the tubulin polymerization to elicit anti-cancer activity. I would like to acknowledge Faculty Pharmacy, Orange, RMC, and my international collaborator. These are my students. These are my publication, my awards, my patent. Thanks for your attention.